these brownies to start the seminar series is not just to kick off the seminar series, but to celebrate the fact that Popular Science magazine has elected our own Maya Breitbart as one of the 10 brilliant scientists in the country. So proud of Maya. We're proud of her every day, but this is just it's gonna explode on Monday.
If you see a photographer, point in the other direction.
So this is just here. Congratulations, Maya. We're very proud to have you at the College of Marine Science. Our new students, so sort of we do TGIFs every other week, typically, and so there will be beer following this seminar, but there's also beer in the Mary Abercrombie Memorial Refrigerator, also Mary is still a graduate student, and that's the beer in the fridge is typically a pay as you feel is reasonable, that, you know, pay as you go.
I am good. Oh, and Chris Songs is going to demonstrate.
Well done, Chris.
You're well on your way to being Popular Science.
I've been practicing.
Well done. The brownies and the refreshments were supplied by the grind. So next time you go into the grind, you can tell Richard how wonderful he is, which we try to do every day.
Yes.
Okay.
Maya has to say something brilliant.
Say something brilliant.
Speech.
Thank you, Jenny. Really?
No.
I'll tell you guys a little story. Actually, I had an interview this morning with the Tampa Tribune, and the guy did what I thought was the best thing a reporter has ever done. And he said at the end, I talked for like an hour about everything, and at the end he said, "You keep saying we. Who is we?" And I said, "I am so glad you asked me that." And then I showed him pictures of all my students and everything, because I think you guys need to remember, anytime one of us gets an award like this, it's really because of the royal we, and I would be nowhere without my lovely lab, who are mostly here. So thank you guys, and everybody else, including including all our collaborators in the room, our wonderful dean, and all of the chocolate that she keeps feeding me, which allows me to continue to be brilliant.
There probably will be more celebrations.
Okay.
But
I never complain about celebrations.
I just thought we'd start with this.
Thank you. Very excited.
And I didn't. Oh, I do have my purse, but it's over there.
She's going to show us how to pay for beer.
The magazine is in Publix if you want to go buy one.
Oh.
Yeah, you're in Publix.
I'm in Publix. That's wonderful. That's
Right.
I know my parents have seen it. That's like the big. My parents found it on their own. That was kind of like the big thing.
That's the big deal.
All right. I guess I'll wait till they get beer. If anyone wants beer or something, now's your chance to get up. Do things.
Should I allow questions during it or hold off on questions?
I usually say either way, and people will interrupt if there's something pressing. If you particularly want them to interrupt, then you can say you can interrupt, but otherwise they'll hit you at the end.
I'm nervous about the YouTube thing now.
Yeah, me too. Just pretend they never told you that, I guess, is the way to go. Oh, I was going to say someone stole my seat, but I can come sit up here. Ben stole my seat.
Yeah.
Perfect. Oh, this is from the seminar panel thing.
Oh, right.
Absolutely. Feel free to move forward. Let me just throw my jacket on this chair so I don't keep losing my seat. I think so.
We'll arrange it nicely.
You will arrange it so
yes.
it's clear that it belongs to me.
Yeah.
That's my brownie too, and my water.
I can't really reach those. Yeah, that's coming up.
Sorry, I did my
my best to mark my space. I promise. Okay. Thank you to everyone for coming to seminar today. Thank you to Jackie for arranging for special treats. I love it here at USF, and it lets me be brilliant, so thanks. But the real reason you are here today, not the brownies, come on, and not the beer either, but is this wonderful man right here, Bob Thacker. So Bob he's at the University of Alabama at Birmingham. He's been there since 2000. He got his PhD at the University of Michigan. He then did postdocs in what I can only imagine are terrible places like the University of Guam and the University of Hawaii. And I guess we kept him up on that humid, really wet weather with this seminar weather here today. So
it's perfect.
Sorry about that. I have known Bob actually because the coral microfield and the sponge microfield are not all that separate sometimes. And he's also collaborated a lot with Cody Sharp at Eckerd. So she's kind of co-sponsoring Bob's visit here. And he's going to tell you about his work on sponge microbiomes, which I've been prepping you all on the micro side all week with BioOsh micro lectures. And welcome, Bob.
All right. All right. Well, it's great to be here today, and thank you for that introduction, Maya. And like she said, I'm going to be talking to you about sponge microbiomes, and we're going to look at a couple different things about sponges as well, and hopefully it'll help you all out. But I don't know if we can get the lights up front because I've got a couple of dark things. Yeah, okay, that would be great. And so we can get started. And so what we do in my lab is really focus on this idea of the evolution and ecology of sponges. And that's what I started working on as a postdoc. I'd actually done all my graduate work on hermit crabs, so it was a big switch for me to go into the sponge world. But in my lab, we work on just general ecology and evolution questions. But that led us into looking at biodiversity because many different sponge taxa that you might want to work with and do experiments with are not described, right? So you have to somehow attach a name to them and describe those species. And then we've also started working on symbiotic interactions with sponges. And that's what I'll focus on mostly today, is looking at these bacterial communities that are inside the sponge. That's led us to do some really large-scale phylogenetic questions that I'll talk to you about as well. I'm the leader of this Periphery Tree of Life, or Portal project. That's also how Maya and I are collaborating by exchanging material for our Tree of Life projects. And with that, doing these really big applications for phylogenetics, we found that we needed to have some more infrastructure. And so I'm also part of an NSF team called Arbor, which is part of their analyzing, visualizing, and assembling the Tree of Life program. Okay, so I'll show you a little bit about Arbor and where we're going with that, but that's fairly new for me. Okay, so what I'll do is I'll give you all an introduction to this whole study system, and then we're going to zero in on one kind of symbiont. I really focus on cyanobacteria, and you'll see why. It's because we can manipulate them. And then we'll look at the ecology of these and look at whether these cyanobacteria could be mutualists or parasites. We'll get into some sponge systematics with our Periphery Tree of Life, or we just call that Portal for short. And then we'll look at these Avital things for this, it's a long thing to say again, right? But this whole thing with Avital, and our project's called Arbor as part of that. Okay, so what's a sponge? So most of y'all probably know what a sponge is, but some people, have y'all ever not picked up a sponge before? Some people have never held them up. Yes, some people have, some people haven't, right? So if you see a beautiful sponge out on the reef, right, you got to think what's happening inside there, right? They look like they're just sitting there. But actually, there's a lot of movements happening inside the sponge, and they're passing water currents through the whole sponge body, right? So the water will flow inside these darker line chambers, or these choanocyte chambers that have these choanoflagellate-like cells here. These collar cells have a little bit of a collar here that will trap bacteria and filter things out of the water, whereas there's a little flagella here that acts like a pump. And if you have all those things moving in synchrony, you can actually move water through this whole body. Okay, but inside the sponge, there's also this area called the mesohyle, the space between all its canals. And that has all kinds of different cell types. So there's many different types of cells in there. Some of them look like little amoebas, these archaeocytes, and they can move around inside the sponge, right? And then there's all kinds of different skeletal structures for sponges. So some have these kind of spicule-like structures. And then when we look at the sponge body itself, there's also a special cell type, these pinacocytes, that line these canals and line the outside of the body and help define like what that sponge is, where the boundary is. But when you look at taxonomy of sponges and really morphological systematics of sponges, you really have a lot of different characters you can choose to work with. There's many sponges will have these kind of silica-based spicules, and some have calcium carbonate-based spicules in them, right? These spicules can be arranged a lot of different ways. If there's fibers present in the sponge, those spicules can project out of it, or we say it echinates that, or they could be inside these fibers. So you could say the spicules core the fiber. And then these could be arranged in all kinds of different ways. So some sponges will have these long spicules holding up a layer of little round spheres, and then on top of that there'll be another layer like a palisade making the outside boundary of that sponge. Okay, so there's all kinds of neat layers and ways sponges are arranged. If you look at these choanocyte chambers inside, you can see these little chambers of cells, then that's where all the action's taking place for these sponges. Okay. So in my lab, though, we use mostly molecular techniques to do systematics, right? We're doing some morphology to identify everything, but we're really trying to place that morphology in a big molecular systematics framework. And so this is a slide from Gert Verheide, and he was able to loan me this one. It's from a paper that's part of our Portal team did in 2009 in Current Biology. But it just really shows you that sponges are this big ancient lineage of metazoan. So you can think about sponges here being near the base of the metazoa tree, right before you have diversification into placozoa there, and then here's all your cnidarians, and then these other groups like Lophotrochozoa, Ecdysozoa, and Deuterostome. Okay? So one of the big findings of our Portal project is that we can really confirm this idea that there is a single evolutionary origin for sponges, okay? And so then you have 4 big lineages of sponges that split off from that. One thing that you might notice in your textbooks, you might still just have 3 lineages of sponges in there, or 3 classes. But this Homo scleromorpha, I'll give you an example of that later, and that's a relatively new class that was established in the last 2 years, right? So you could think, well, okay, why would you want to study all these sponges besides this really neat evolutionary history that they have? One big idea is that they're really diverse. There's about over 8,000 species that are accepted names of sponges, but we think that about 15,000 species actually exist. Okay, and you can get to this number a couple ways. One is if you went around to different museums in the world and just looked on the shelf, you can say, well, what things are actually cataloged and have a name with them, and what things just say SPA, right? And nobody's put a name on there. And there's about 5,000 things in museum collections worldwide right now that just say, sponge X, right? And nobody's actually taken time to put the name on it. So that's one thing we're trying to do as just a general community of sponge biologists through our Peripheral Tree of Life project, is to get some of these things described and attached more names and things like that. But we also try to discover new species. And so in just one dive in Bocotea, Panama in 2010, we found 10 undescribed species just all at once, right? And so every time we go to new kinds of habitats or little microenvironments, you can find new species of sponges very quickly, okay? And so we also know there's this big rate of discovering new things. Okay, so just to show you, these are 4 different new species of sponges from Panama, and I have to say I've been remiss. We still don't have names for these, even though we picked them up about 2 years ago. So it still takes a long time to get these descriptions done. But these 2 are relatives of your Zestospongia muta, the big barrel sponge that's out on the Caribbean reef. This one is a type of Dicydia, and this is a calcareous sponge here. Okay, another reason that you want to study sponges is for biotechnology purposes, right? So sponges are really important sources of pharmaceutical compounds and other types of natural products. You've got an expert here, Bill Baker, over here in the front on those. And this is just one example of Tectotethia chiensis. It looks like really drab for a sponge. I mean, you can snorkel right over it and not notice it, and everything's in seagrass beds in Panama. But you can get cytosine arabinoside from this sponge, and that's a drug that's actually used to treat leukemia. So that's a really good source of specimens, or source of these pharmaceutical compounds. But for us, a big source of specimens has been working with the National Cancer Institute at the National Institutes of Health. They've actually loaned us a lot of material. They've let us process that for both morphological analyses and molecular systematics. So it's been a huge source of material for our project. Okay, so this also brings up this idea of sponge microbiology. So a lot of those compounds that can be isolated from a sponge aren't made by the animal itself. Okay, so they're made by bacteria that live inside the sponge. And we know that some sponges have just tremendously abundant and diverse communities. This on the left here, this is a TEM from Nicole Webster, and it's a slice through a sponge called Rhophiloides. This is just in the middle of the sponge in the mesohyle, and all the cells that you see here are bacterial cells. Okay, so there's no animal cells or no sponge cells in that picture, right? So that's pretty wild. So you think, how does this thing exist with this huge density of bacteria inside it? On the right here, this is a micrograph with the confocal I made with Cody Sharp's help. We actually made this, and I mean, I was on sabbatical one year. But this is a sponge called Herdios violacea. It's just a histological slice through it. And what's lighting up here with the stain is just a general eubacteria stain. Okay, so all these areas where you see it lit up here on the edges here, those are all just bacterial cells living inside of the sponge. Okay. So one thing in my lab that we try to do is to think about these symbiosis with microbes inside that sponge in a really general theme of symbiotic interaction. Okay. So we want to think about this like we would, say, if you were studying soy, right? And you're looking at the rhizobia living in these root nodules in a soy plant. We also try to think about it in terms of corals and the zooxanthellae that live inside of corals, right? So we're trying to draw on some general ecological principles and think about what controls the diversity of these things, what controls their abundance, and then do they actually have a benefit to their host, or do they have some kind of cost to them? Okay, so that gives you kind of an introduction for what I study and things. And so now what we're going to do is try to look at the diversity of these interactions in terms of just their cyanobacteria and what kind of cyanobacteria live inside a sponge. And you're going to see that there's really just two kinds that we're going to talk about today. There's the filamentous one that is about 10 micron wide filaments that are anywhere from maybe 10 to 50 cells long in that filament. Then there's also a unicellular one that's about 500 nanometers, 800 nanometers across. The filamentous one is called Oscillatoria spongellii, and the unicellular is Synechococcus spongiarum. So they all have the sponge in the name, right? So you can think of I'll just call them one Oscillatoria and the other Synechococcus, okay? So when you think about this diversity, this huge amount of bacteria that could be inside that sponge, you could approach it a few different ways, right? So one way is what I like to call a microbial perspective, and that's when you might take a sponge and take it back to your lab, put it in a blender, right, extract all its DNA, or put the whole thing out on culture plates and see everything you can culture from that sponge and all the DNA you could isolate from that, right? And if you're doing that kind of approach, you can actually come up with a huge diversity of microbes that you found associated with the sponge. This is an image from a paper by Mike Taylor that he published in 2007, where he compiled thousands of different sponge-derived 16S ribosomal gene sequences. And he found that representatives of 16 different bacterial phyla and both archaeal lineages are reported there as sponge symbionts. Okay? So this was like a starting point and a kind of a summary in 2007 of everything that people had. And from that, this whole field of sponge microbiology has just exploded in the past 6 years. We're now part of this giant project with the Earth Microbiome Project, where over 1,000 sponges have already been submitted to that project, and they're trying to categorize almost just even deep sequencing an even broader diversity of microbes out of those hosts. Right? But in my lab, we kind of take a different perspective most of the time and take more of a host perspective, because I am still coming from the side of a sponge biologist more instead of as a microbiologist, I guess. And when I call this host perspective, what I mean is that we'll just look at a lot of different host sponges, but we'll just try to find one symbiont and see if that one symbiont is in all these different hosts. Okay? And our targets are usually cyanobacteria, and that's because it's really easy to index how abundant they are, right? We can just use the chlorophyll A concentration in that sponge to figure out, are there going to be cyanobacteria here or not, right? And that's as simple as taking a sponge, whacking off a piece of that, sticking it in some acetone. Does it turn green? And you can say, okay, yes, let's keep going. Right? So that makes it's a really easy way that you can can narrow down your field of study and find the things that you want. Okay, another thing we can do with those is identify these symbionts really quickly, because we have specific PCR primers to pull out those symbionts and identify them. So most of the work I'll show you today takes place in Panama. We work out at Bocas del Toro. It's right near the border with Costa Rica on the Caribbean side. And in Bocas, there's a big island that's Isla Colón, where the Smithsonian Institution has a marine station. So it's right here at this little narrow point in the island there, right by the Bocas town. So we've done a lot of work there over the past few years, and I actually teach a class on sponges there. We offer it about every 2 to 3 years, where we just do sponge taxonomy and ecology. But if we're in Bocas and you wanted to survey a lot of these sponges, one thing you could do is just try to get every sponge you can. And this is looking at 60 different species we found, and we measured how much chlorophyll A was in that sponge. Okay, and this is in micrograms per gram of the sponge. And you'll see that there's a whole set of sponges here. It's about 20 sponges, about a third of them, that have over 150 micrograms per gram for chlorophyll A. And as a comparable amount, that's what you would get in some grass if you measured it out here, right, or some trees, that if you look at the leaves of those trees. Okay, so it's a really large amount of chlorophyll. We had 2 intermediate things here, and then those were sponges that we later found out those were what we would call bleached now. They were sponges where they have photosymbionts, but they lost a lot of those. They're in a really hot environment. Then all these ones with the dark bars, those are things where there's just a background level of chlorophyll A that seems to just be from phytoplankton that's passing through the sponge that the sponge is eating. Okay? But so with those 1/3 of the reef sponges having these cyanobacteria in them, we wanted to know how abundant they were as well. So we did a lot of reef surveys. This is summarized here on the bottom. It's the percent sponges of our total sponge community out of a huge survey of reefs around the Bocas del Toro area. And so you can see that even though these things have about a third of the sponges have these photosymbionts, they're not the most abundant sponges out there. They actually account for about 1/3 of your total sponge abundance as well. Okay. So right away you should have a question in your mind, right? Well, if these are so great, it's so great to have photosymbionts, why aren't all these bars for their abundance like way up here? Like why aren't these things just taking over the whole reef, right? So that's immediately why we thought there might be some cost to hosting these symbionts. All right, so we want to take a look at symbionts in a little bit more detail, and we can start with these Oscillatoria. And so with the Oscillatoria, we've got 3 really common hosts in the Caribbean. There's a Haliclona, a Zestospongia, and a Herdios. And then if we go out to Guam, that's where I did my postdoc work, there's some Lamellodicidia species, Philospongia, and Dicidia that all have these Oscillatoria in them. Okay? So it's a pretty wide diversity of sponges that are hosting these. All of these ones in the Pacific are in one group, the Dicideidae. And then these ones that are in the Caribbean are in this Herdios is actually now transferred to Lamellodicidia from some recent work we've done. But these Haliclonia and Zestospongia, they're in a completely different order, the Haploscelerida. Okay, so it's 2 different orders hosting these things. So our procedures are pretty simple with these. We go out and we collect a sample, and this is Cristina Diaz. Some of y'all have met her. She works with me a lot in Panama. She's collecting a sample out of this seagrass bed. And then we bring it back to the lab. This is one of my students, Zolchi Morgan. She's taking the sponge sample and then making a total genomic DNA extraction there, right? So we call that our metagenomic extract because we know we've got the sponge sample and then all the bacteria that are inside of that, right? What we can do then is use some PCR to amplify the sponge nuclear ribosomal genes and the cyanobacterial ribosomal genes. And that's how we kind of separate everything out in terms of getting a phylogeny for sponges and a phylogeny for their symbionts. So just to give you a quick overview of these Oscillatoria, I could give you all a whole talk just on that. I thought I'd just give you all just a smattering of different things today. But with these Oscillatoria, we found that there's one specific clade of all these symbionts, okay, that's related to some free-living strains that we have associated with the disease of sponges, this Aplycina red band syndrome. Also, we have this free-living Lingbia that's forms mats in Guam. That's also a close relative of this Oscillatoria. But within our sponges, each sponge that we've examined has a unique genotype, you could think, or a unique strain of that cyanobacterium associated with it. And we've never seen a mixture inside any host species there. Okay, so we have a symbiont for Dicidia granulosa here, one for like Philospongia papyracea or Zestospongia bocotorensis, and they never seem to have a mixture of different types of symbionts in those sponges. So because we had found that out, we thought, well, maybe this means that these things are vertically transmitted, right? That would be giving you some really good evidence for that. But we have one caution with that is that we know that, say, these ones in the Zestaspongia and Haliclona are in a completely separate order of sponges than all those other ones, okay? And in that order, there's hundreds of sponge species, so why do just 2 things in that order have those, right? So we also know then these have to have some ability to horizontally transfer and infect new hosts and things like that over time. So we thought that this might be vertically transmitted, and then finally we were able to show that in 2010. The sponge here, this is Zestaspongia bocotea. It's only found in Bocotea as far as we know. We've been trying to look for it in other spots in the Caribbean, but haven't found it anywhere else. This sponge, if you peel it off this rock that it's growing on, you can look underneath it, and it'll have a brood chamber here. And inside that brood chamber, these developing embryos are all different sizes and colors, right? So you can tell that they're developing asynchronously, right? So they're all at different stages of development there. And these darker ones have been already infected with this cyanobacteria, okay? And you can actually see this happening if you look in the microscope, right? So you've got adult tissue around here, and here's some embryos. And you can see the filaments just moving into these embryos and colonizing them. And then what's really neat is when these larvae hatch out, they look like this. These larvae come out and they start swimming around. And this is just epifluorescence, right? I'm not staining that or anything. It's just the chlorophyll A that's lighting up here. And you can actually see the boundary of the larva right there, okay? So when this larvae is swimming around, it's just like a bag of these cyanobacterial filaments, right? And I keep thinking, how is it still an animal and it's swimming around and functioning as a sponge larva, even though it's just packed out with these bacteria? Okay, so to summarize for these filamentous cyanobacteria, it's Oscillatoria spongeli. There's a lot of different clades. So every host that we've looked at that has that Oscillatoria in there, it has a unique strain, right? And we don't see any mixing of those. We have observed in vertical transmission, right? And we have, yeah, sure, yeah, sure, sure, yeah, sure, yeah.
I study volcanoes.
Yeah.
Vertical transmission
yeah.
means from parent.
From like the parent to the offspring. Sorry about that. Yeah. Right. Right, so so like this idea here, where you have a parent and its symbionts are infecting its offspring, right? Well, you can say between adults, or like if this was out in the water column and these just get acquired from the water, right? So that's a good question, though. Okay, so but so if you think about this idea that they're able to pass it to their offspring, right, and that's how these symbionts get transferred. That idea that you have vertical transmission, and over evolutionary time, then you're going to think that it's going to be a specific genotype associated with that species, right? If that's really tight and you don't have any other form of transmission occurring, right? So that's what we have evidence for in this group. But we don't really have evidence for co-speciation. So I'm not showing you all the sponge phylogenies for these groups, but the reason why we have really weak evidence, we think, is that there's tons of sponges related to all these ones I've shown you that don't have the symbiont, right? So we don't know how that initial infection happens, right? And then it's persistent in one species over time, but it doesn't seem that we have a lot of speciation within those groups. We can't see, say a whole group of things like, say, with figs and their fig wasps associated with them. We don't see anything like that with this huge amount of diversity happening because you have these symbionts, okay? We do have, though, evidence that there's a lot of times over evolutionary time that different sponges have been colonized by the symbiont, right? Okay, so let's look at these unicellular Synechococcus. And so if some of y'all have done more open ocean type things with phytoplankton, you know that your Synechococcus are one of your dominant photosynthesizers out in the open ocean, right? But our view on this one is that we first thought it was a generalist that was in every sponge we were looking at. And now we know it really has a lot of specialist genotypes, okay? And it's really similar to what's happened with looking at the zooxanthellae inside of corals, right? So with the zooxanthellae, the Symbiodinium, originally it was thought it was a real generalist in that there was the same Symbiodinium in all these corals around the world, right? And now you know that all these different corals and different habitats will have different clades or strains or genotypes of that Symbiodinium. So we've seen the same kind of phenomenon as we've studied these Synechococcus. And so just to let you know, just around here and in the Caribbean, you have several species of Varangids that have these. Things like Aplycina fistularis are common in the Florida Keys, or Aplycina fulva. Then also Neopetrosia subtriangularis is really common in Florida, but also Zestaspongia muta. This is the big barrel sponge you get out on the reefs, right? So when we first made a phylogeny of this group, we used our 16S sequences for ribosomal RNA, and we could compare it to all these different strains of free-living Synechococcus that are all out in culture. We found that our sponge-associated Synechococcus formed one big clade, and that there is no genetic differentiation within that clade. Okay? We also tried to culture these, and it turned out there's labs all around the world trying to culture these Synechococcus, and no one has succeeded yet. Okay, so there's a lot of people still trying even now. I've been hesitant to put students on that as a project because, you know, if no one succeeded in 25 years of trying, I don't want to say you've got to get your master's in 2 years and, you know, be done. You know, so that's a tougher one to do, right? It's a tougher project. So we have been working a lot with people that study open ocean Synechococcus, and Gabrielle Rocap let us in on how she was trying to differentiate different strains of Synechococcus. And that was using this ITS, this internal transcribed spacer that sits between the 16S gene and the 23S ribosomal subunit. And luckily in these cyanobacteria in the Synechococcus, there's two copies. You got to tell them apart, but one of the copies has 2 transfer RNAs sitting there in the spacer region. And that's super handy for doing molecular work because you can base your primers off these tRNAs. You can always check to make sure you've got that copy that has the transfer RNA in there. It makes it a lot easier to know that you're getting the right sequence out of the right organism because all these are coming from environmental samples, right? So when we did that work, we found that, like with our Symbiodinium, these Synechococcus have at least 50 different clades. This is too impossible for anybody to read, right? But there's these are like A all the way to K, and we've got now a bunch more. Like we're down to like Q, R, and S or something by now. So there's lots of different clades of these. And some of them are very specific, and they'll just be in, say, one genus or one species of sponge. But others are in mixtures. And so one thing you can think of with these is your Aplycinas around here, like the Aplycina coliformis and Aplycina fulva, they have a mixture of clades A and B. And actually, I added my slide because a student in my lab just a couple weeks ago ran another set of DGGs on these, and they can detect a clade C in these as well now. These Neopetrosia, we know, have clades B and C in them, right? But when we've done a big analysis of molecular variants on this data set, we find a really significant influence of both the host phylogeny. So we know there's this some sort of co-speciation you could think of happening, or specialization to different families or genera of sponges. But we also know there's a huge influence of geography. So you find different clades of the Symbiodinium when you're up here in Florida versus if you're in Panama versus if you were in the Mediterranean, right, or in Indonesia. Okay? So we know there's this odd combination here of an interaction of both the host biology and the host phylogeny and where in the world you're located and the biogeography of these symbionts. Okay? So that's something that we're trying to tease apart a lot more now. Okay, with these Synechococcus then, the thing to remember with these is that there's a lot of different clades now we think of. We used to think this was just one thing, but now there's about 15 different types. We know that distribution among sponge hosts is influenced by both the geography and the phylogeny of the host. There is vertical transmission of these. Okay, we do have one observation from a lab in Australia where you can make an SEM of the developing embryos in one sponge species. It's a chondrilla species. And in the eggs, these Symbiodinium are being transferred from a maternal cell, which is the cell that provisions that egg and lets the egg develop. You can actually see a little stream of cytoplasm connecting the cell to the egg, and there's Synechococcus moving through it, right? So we know that can happen. But we don't really have any evidence in our genetic evidence when we do a phylogeny of that, right? So our phylogenetic evidence is for this horizontal transmission and for a lot of transmission due to the environment and things picking up a local symbiont that's really maybe adapted to that local environment, right? So this is still a big puzzle for us in this case because we don't really know how this transmission happened. A bunch of those sponges I just showed you, no one's ever seen larvae, right? So it's like, how do we know how they're reproduced or how the egg develops and things like that, right? So that's where we're in a puzzle with these. Okay, then what we spent the bulk of our work in the lab in the past maybe 5 years doing is trying to manipulate these symbioses. And because they're cyanobacteria, we thought we'll focus on those because we can just put shade cloth over the sponge, cut out light, and then see what happens, right? So it's a simple way to kind of manipulate the whole system. And what we're looking to do here is that we think the benefits of these cyanobacteria are that they're doing photosynthesis, right? They could be providing carbon or even nitrogen to the host. We know that some of them make secondary metabolites that defend the host from, say, predation, right? Or maybe from other sponges growing on top of them and things like that. And any of those kind of benefits would indicate this is some kind of mutualistic interaction, right? But we think there's a cost here because not all sponges have these. And so that cost, we think, is cyanobacterial growth, right? And there's actually one published observation by Klaus Rutzler in Belize where he saw the Symbiotic type sponges or symbionts overgrowing the host sponge, okay? And the sponge is actually becoming more and more green over a period of weeks, sloughing off tissue. And in his words, he was thinking that the sponge looked like it was trying to get rid of these symbionts as fast as it could, right? But it was somehow failing to do that. So we think that if sponges can't control the growth of those cyanobacteria, these could indicate that there's some kind of parasitic interaction happening. Okay? So we wanted to try to see, do sponges really benefit from these interactions? And if we shade the sponges, we can reduce their photosynthetic activity, right? And then we'd remove whatever host or whatever harm or benefit would be happening here. So our idea here was that if we saw less growth when we shade these sponges, then that would indicate that they're mutualists. If there's no change, they could be commensals, right? And if there's more growth, maybe these are parasites. Okay? So here's how we kind of set it up. This is kind of an overview of a reef in Panama where we've got these set out, and we've got some dark canopies over a grate and some transparent canopies. And so here's like one of these dark canopies. This is all just PVC and cable ties, and these egg crates are things like you could get for your fluorescent light covers. Okay, so these are really Home Depot oriented supplies and stuff. You can just cable tie sponges in here, leave them out for 8 weeks, 12 weeks, and then harvest them again at the end of that time. And so this is just a shot down through one of these clear canopies that we have. So after you do that the kind of experiment, here's some results. And this first one is with Lamellodicidia. The middle one here is from a Neopetrosia. With Lamellodicidia, I put the little figure here. It has Oscillatoria in it. The Neopetrosia has a Symbiotic in there. And on the far right here, this is Aplycina fulva, and it has a Symbiotic in there. Okay? So this top set of graphs is with our experiment looking just at the change in chlorophyll A during the experiment. Okay? And always on here, it's going to be this lighter bar is the control, and then this darker bar is the shaded experiment. Okay? So with Lamellodicidia, for example, you can see that there's no change really in the relative amount of chlorophyll A present. Okay? And this is, again, you can think about this as the ratio of chlorophyll or the ratio of cyanobacterial cells to the sponge cells. Okay? On this bottom set of graphs, though, this is the change in dry mass in those sponges. Okay? And so for these Lamellodicidia, you can see that for the controls, there's a little bit of growth. This experiment only went for 2 weeks because at the end of 2 weeks, we've lost 50% of the mass in these shaded sponges. If we go to 3 weeks, which is the way we first ran the experiment, right, all these things disappeared and we didn't have anything to measure, right? So in about 2.5 weeks, these sponges will die if you shade them, right? So that told us that this might be a really obligate mutualism, right? If these sponges don't have access to light, they're not going to survive. Okay. For the Neopetrosia and this Aplycina, what's interesting is if you shade these, they lose about half of their symbionts in the shade. These Neopetrosia actually gained a lot of chlorophyll A in the control conditions. We realized that our canopies actually block UV light. And so some sponges do better, and some symbionts seem to do better under that UV blockage. Okay. But look what happens here at their dry mass here. If you look at the Neopetrosia, it's even here, right? There's no significant difference there. It doesn't matter if they lose those symbionts or not. That sponge grows at about the same rate. Okay? So we can't measure any impact of these cyanobacteria on that sponge, and we think that they really are maybe true commensals in there. Okay? For this Aplycina, though, you can see that this sponge grows really well. This is over about 8 weeks. You can see about 60% increase in mass in the controls, and it's about a 30% increase in these shaded ones. So when you knock down these symbionts, you knock down growth. It doesn't really kill it. So we think this is more of a facultative mutualism. The sponge will do a lot better with those symbionts in place, but it doesn't do very well, or it does less well, you could think of, when those symbionts are reduced. Okay, so the things to take out of that then are the shading we know really decreases the abundance of these symbionts. this Oscillatoria seems to form an obligate mutualism. And I didn't show you all the other experiments we've done with this, but we've used about 5 different species that host Oscillatoria in shading experiments. And in these conditions, the sponge cells and the cyanobacterial cells seem to die at about an equal rate for these. Okay? For this Synechococcus spongiarum, it appears to form a range of interactions. And so we find this one really the most interesting because they go from commensalism to facultative mutualism. And we think we have some evidence from one of my grad students this past summer of a parasitic interaction with these Synechococcus as well. Okay? So we think it's forming a range of outcomes, you could think, with different types of hosts. So now our big question is, well, is that due to the genotype of the symbiont, right? Or is there something about the host physiology involved with regulating that interaction? Okay, so one question that we had coming out of all that was that, well, could our host actually cope with the loss of these Synechococcus, these ones where we don't see growth and we think that's a commensalism? One of my grad students actually thought of this. It's like, couldn't they just increase their heterotrophic feeding rate and just make up for the loss of photosynthesis and then they're okay, right? And they can just stabilize their growth. So that meant, well, can we really estimate that? Can we measure heterotrophy and phototrophy with these sponges? And more than that, can we really track nutrient transfer from the host, from the symbiont to the host or the host to the symbiont? So Chris Freeman, one of my students that just graduated last year, he's now a postdoc at the Smithsonian Marine Station over in Fort Pierce. And he had this great idea of using stable isotopes to trace the flow of nitrogen and carbon during our experiments. And so he set out much more complicated experiments out with these shading cloths. And you're going to see some later on where these are different amounts of window screening, so we can vary the intensity of shading. We can get a big gradient of that through our experiment. But the key things to remember is that these isotopes for carbon and nitrogen, these are a little bit heavier isotopes, and they become more concentrated as you move up to higher trophic levels. Okay, so they're really commonly used in big food web studies, right, and trying to look at trophic interactions. So what Chris did was to couple a shading experiment with these kinds of stable isotope measurements. And in this case, it was pretty easy to take our sponges, dissociate all the tissues, and then we can use differential centrifugation to separate the cells into microbial cells and then into animal cells. Okay? And just to give you a little refresher on stable isotopes, I'm going to show you some axes like this with our delta N15 going up on the y-axis and delta C13 on the x-axis. And in both cases, if you see trophic enrichment, you're moving up the y-axis or across to the right on this x-axis. Okay? And just for an example, if you measured, say, particular organic material out in the water column, you'd have like a palm circle here. If that's being consumed by a heterotroph, that heterotroph should be accumulating this and transferring this or accumulating more of this heavy C13 and this N15. So let's look at a sponge, and this is our control sponge, Niphetes erecta. We like to use the sponge as our positive control because it always grows. We have a hard time killing it, right? So that's a good positive control. And then it doesn't have very many bacteria in there. It has some bacteria in there, but it's not a hugely dense bacteria population. Okay? So when we look at this one, we can say, here's our particular organic matter that was out in the water column. And then when we sample either the sponge under shaded conditions or under control conditions, these would be the sponge cells, or the microbial cells under shaded or control conditions, all these are jumbled up on top of each other. Okay, so this would kind of confirm that this sponge is that heterotroph feeding on this particular organic matter, and that it also hosts some heterotrophic bacteria in it. Okay, so the bacteria you get out of that are mostly heterotrophs. When we look at Aplycina coliformis, if you look at the microbial fraction and just control conditions, it's way shifted down here. Okay, and this is more like what you would see with an oceanic Synechococcus. Okay, when we look at the sponge, it's feeding just on those symbionts, right? So if it was mixing in more of this particular organic matter, this dot for our sponge cells should be way up here, more to the left and higher in the graph. Okay. Then when we do our shading experiment, something interesting happens. This whole interaction between the symbionts and the sponge shifts over to the right. Okay, and if any of y'all are good at cyanobacterial biochemistry, we'd like your help in figuring out why that happens, because we still haven't figured that out. But we know we've somehow impacted with the shading how carbon's being processed by the symbionts, but that's still the main source of carbon and nitrogen for the sponge here. Okay, so that's our best example yet of there being a really tight interaction between the sponge and the cyanobacteria here. Okay, so one way we could think about these then, you can put a number with the mixing model on these, and Niphetes erecta gets about 75% of its carbon from particular organic matter, but Aplycina coliformis gets about 75% of its carbon from microbial symbionts. Okay, so we know that that's happening. And we expected our shading experiment really to disrupt that symbiosis. We thought we would just see that all shift over in the coliformis. They would just start feeding heterotrophically, right? But instead, it's still feeding on its microbial symbionts. Okay? So what we're trying to now figure out is what happened with that carbon metabolism. And that set up another question for us. Did the symbionts actually change? Okay, so with corals and Symbiodinium, one of the big questions people have is with increasing seawater temperatures, are corals going to be able to swap out what type of a genotype of Symbiodinium that they have, get maybe a more heat-resistant clade in there? We're wondering the same thing. We went into shade, we went to a low light condition. It's not completely dark, right? But could these symbionts swap out in these sponges? Okay. So another thing we're trying to figure out is that can we measure actually these costs of hosting the symbionts? Now that we can look at our carbon transfer and nitrogen transfer in terms of these stable isotopes, we want to try to put a number on how much energy is being transferred, right? Or how much benefit can we actually quantify here? What we were thinking that we might have some curve like this for our benefits, like our benefits max out at some certain symbiont density, and that our costs would increase as you get increasing symbiont density in the sponge. Okay? So that we might have some kind of optimum time here where you've got some optimal symbiont density where you've got the most benefit for the least cost to that host. Okay? So you can think you got this net benefit curve like this. So one way we wanted to try to do this is thinking about how we can manipulate that symbiont density. And the one way we can do it right now is by shading, right? So I showed you how we could knock it down in half with certain levels of shading, right? So what we decided to do is do a whole gradient of shade cloth. And basically we just use window screening that we can do in one layer all the way up to, say, 10 layers out on a reef, and then figure out a bunch of replicate sponges to put under that window screening. And our idea was that we would be able to have some intermediate level of shade where we would be able to see the maximum growth rate of these sponges. So we used two sponges that really grow well in these experiments, these rope sponges, Aplycina coliformis and Aplycina fulva. We used 6 different irradiances in the one I'm going to show you here, and we kept the sponges under those for about 11 weeks, and then we measured the relative growth rates of these sponges. Okay? So for Aplycina fulva here, this is showing you its growth. It does the best when there's no shade over it at all, and then it just drops off immediately. Okay, so as soon as it has a little bit of shade over it, this drops down, and then it stays at this low level of growth. For Aplycina coliformis, we get something different. We get, with a little bit of shade, it actually does a lot better. Right? And then as you increase the shade, it drops down, and then it comes back up again. Okay? So that's where we found something really wild. We had to go back through and check this again. The students who did some of this work, I said, okay, we're going back to everybody's notebook, and we're reanalyzing our data again, right? And we made sure, but sure enough, we get this trend of it coming up, it coming down, and then it comes back up again. Okay? So again, you have to think, well, what's happening to metabolism during this type of experiment? Or are our symbionts kind of switching out during this? So what we've done so far is to figure out that our different host species do have these unique